Good evening, one and all. Hello. Good evening, one and all.
Good evening, sir.
Good evening, sir.
Is it audible to you?
Yeah. Yes, sir.
Yes, sir.
Right. What is our last class?
Sir, protoplast.
Culturing somatic hybridization. Somatic hybridization, protoplast culture, hybrids. In last class, we learnt about somatic hybridization rather than sexual process involved by fusion of two vegetative cell protoplast. You know what is protoplast, where it is. And you know how to separate the protoplast from the vegetative cells. What are the methods to separate protoplast?
Enzymatic method. Mechanical.
Mechanical, enzymatic, right. So any method, we separated the protoplast, we culturing the protoplast on agar nutritional media. After that, transfer this into tissue culture technique. So that's what we learned in last class. And also applications of protoplast culture and hybrids also learned. Today class, transgenic plants. What is transgenic? Transgenic means the name itself given transfer. Transfer what here? Hello?
Audible, sir?
Clear?
Yes, sir. Everything is clear.
Right. So what is transgenic? Are you able to see these diagrams?
Yes, sir. Visible.
Right.
So genes are transferred.
Right. Genes are transferred from where to where?
One organism to another.
Right. Genes are transferred for one organism to another organism. Just wait. So this is one plant. And this is another plant. So do you want to transfer the gene from one organism to another organism through the vectors? First of all, you selected the gene from this one. This is A. Suppose this is B. Transfer the gene, G for gene, and inserted into another plant to get desired gene without culturing. Most probably in this process, no culture. Through vectors, they are injected into the another plants. Now what happened here? That gene transferred from one plant to the another plant, one organism to another organism. Later they inculcate this all desired character of the A plant, and these characters obtained in B plant also. In general, the transfer of gene takes place in meiotic division between the homozygous chromosomes or homologous chromosomes. What is the plant body? What is the ploidy of most plant cells or most animal cells?
Diploid cells.
Diploid, sir.
Right. Diploid cells. The diploid cells means this is blue color. You might be seeing this blue color gene and red color gene. These are formed as a pair, right? So this pairing called as diploid state. In this pair, one from mother, that is one for female, because considered as another one male. But during crossing over in meiotic division, during crossing over means we can observe here this region. During crossing over, exchange the gene. That means transfer the one segment to the another segment. Blue to the red, red to the blue. That's why you can see here the exchange, transfer of genes. Transfer of genes. So this blue color added the red gene and red color added blue gene. Now these are separated. That means becomes haploid. Becomes haploid. During fertilization, these haploid gametes fused. What happened? Some new characters obtained. Is it okay? Hello?
Yes, sir.
Yes, sir.
Actually, first completely blue color chromosomes, red color chromosome. What happened after crossing over? The exchange, the transfers of gene, blue to red, red to blue. That's why next progeny, the new characters obtained. It might be desired character or undesirable character. It may be beneficial or unbeneficial to us. But in transgenic process, we selected desired gene and transferred desired genes only from one organism to another organism. The clear definition of the transgenic plants here, just wait. Let me show definition. Right. Are you able to see? Hello?
Yes, sir.
Yes, sir. No, sir.
Clear?
We can see, sir.
Yes,
Okay,
sir.
right. Please write down this definition. You always ready with your notes and pen. Okay? Right. First of all, we have to develop the definition concept. After the, what are the transgenic plants and how they are formed? What are the techniques involved in? But before that, let's see the definition. As we discussed, the transfer of desirable genes. Clear? The transfer of desirable genes. Transfer of desirable genes from one organism to one organism to another organism. One organism, actually not seen here. One organism to another organism by using rDNA technology. No need to write these all things. The simple write transfer of. Are you follow me? Hello?
Yes, sir.
The simply write down the transfer of desirable gene from one organism to another organism by using rDNA technology. Simple word, rDNA technology. We will discuss later what is rDNA technology, what are the techniques involved in. By using rDNA technology, we have got more beneficial plants. More beneficial plants. The plants developed from integration of foreign gene. Integration of foreign gene. Integration of foreign gene. The plants developed from integration of foreign gene are known as transgenic plants. Are known as transgenic plants. Are known as transgenic plants. Known as transgenic plants. So this is the definition. Simple the transfer of gene, right? Through DNA technology, rDNA technology. rDNA means recombinant DNA technology. Just before we saw the crossing over, that is exchange of genes. The exchange of genes nothing but recombination. Combination between two homologous chromosomes. That is recombination. As a result, formation of recombination gene, recombination DNA. So this is called rDNA, right? So the first transgenic crop approved for sale in United States in 1994. The first time transgenic crop approved in US in 1994. The plant name flower saver tomato. Flower saver tomato. So main purpose to introduce this plant, longer shelf life period, to create shelf life period. What is shelf life? Hello? What is shelf life? So in general, how many days the tomato in general, we must have seen the so many tomatoes in our daily life. How many days they are in fresh? Hello?
Yes, sir.
Do not get rotten. How many days they are in fresh in and always in unripened position?
Okay. 5 to 6 days, sir.
So 5 to 6 days. Okay, 1 week.
Maybe short duration.
Yes. But in longer shelf life, they can appear in fresh up to 2 to 3 months. That is main concept to produce flavor saver tomato.
Yes, sir.
Right. This is the first plant introduced in USA 1994. The next, what is rDNA technology and genetic engineering techniques? Let's see rDNA technology and genetic engineering techniques. In our book given the page number 238, genetic engineering techniques, but it is given very short. But rDNA technology very important to understand about transgenic plants. So please follow and write down the class. First, rDNA technology. Write down rDNA technology or genetic engineering techniques. Write down rDNA technology or genetic engineering techniques. Are you ready?
Yes, sir.
Right. Already told you what is rDNA. That means recombinant DNA. Right?
Yes, sir.
Already we seen in natural process, the transfer and selection desired genes are time consuming. That means in meiotic division, in natural process, this recombinant DNA formed. Isn't it or not?
Sir, can you repeat once again?
In general, this rDNA formation, that means recombinations occur in naturally during meiotic division in plants in the form of crossing over. Already we have seen crossing over process. Should I play the slide again? Hello?
Yes, sir.
The simple what we learned in just before slide. Let me draw. Red color, blue color gene, these genes are exchange each other. And as a result, formation of recombination means here the red color with blue color. So this is recombination form. Already told you one sentence. It is very difficult to identify the desired gene in this combination. So these recombinations as exactly desired genes or other gene. We are unable to decide this whether it is desired gene or undesired genes. This problem of gene transmissions are overcome with the discovery of new technique popularly known as recombination DNA techniques or recombination DNA technology. The main essence of this technology has been to transfer specific gene from one organism to another desired organism or target organism, and desired product can be scored. Desired product can be produced. So recombinant DNA technique was introduced in 1970. Recombinant DNA technology introduced in 1970. Just wait. Recombinant DNA. Right. Are you able to see? Hello?
Yes.
Yes, sir. Yes, sir.
Yes, sir.
Right. rDNA technology. It was discovered in 1970. 1970 by Stanley Cohen. Write down the name Stanley Cohen, Stanley and Herbert Boyer. Stanley and Herbert Boyer, Boyer. Stanley and Boyer. So let's see the basic concept in this, or basic steps in rDNA technology. Another name of rDNA technology is gene cloning. Remember, recombination gene, rDNA technology, and gene cloning. Another name. Gene cloning. Right. So in rDNA technology or gene cloning, there are some harder steps. These are in first one write down. Identification of desired gene. First, we have to identification of desired gene from the donor cell. Right. identification right. First step, identification of desired gene. Identification of desired gene from the donor cell. This is the donor cell. This green color desired gene or foreign gene. So first of all, we have to identify. That is the first step. Second, after identification, first one, identification. After identification, next, isolation of desired gene. Second, we have to isolate desired gene from the donor cell. Next, this isolated gene inject into appropriate vector. So here the bacterial cell as the vector. Bacterial cell plasmid, not exactly plasmid, bacterial cell plasmid act as a vector. Now, desired gene selected and vector selected, now join together. That means desired gene and plasmid vector DNA both are joined. In next step, transfer of rDNA into competent host bacteria cell. I will explain later. First remember. So in next step, transfer of rDNA into competent host bacteria cell. Next, selection of bacteria cells that are having rDNA. Selection of rDNA bacteria cell, right? Selection of rDNA bacteria cell. Next, multiplication of rDNA. Multiplication of rDNA. Multiplication of rDNA. Finally, transfer of rDNA into target cell. Transfer of rDNA into target cell. Are you follow me?
Yes, sir.
Hello?
Yes, sir.
Yes,
Right.
sir.
Sir, afterwards, send the screenshot to the group, sir. Drawing.
Right. So what is the first step?
Identification of desired gene from donor cell.
Right.
Sir.
Identification of here we can see that gene green color what gene. So that is the identified first. So this identification gene separated from the donor cell. That is the second step. That means isolation. But here remember the isolation of desired gene by four different methods. These four different methods are employed for isolation of DNA. One is mechanical shearing, cleavage by restriction enzymes, RNA directed synthesis, and chemical synthesis. Just listen, do not write all, but we follow only cleavage by restriction enzyme, only one method. Actually, there are four methods, remember it. Mechanical shearing, cleavage by restriction enzymes, RNA directed synthesis, chemical synthesis. In this, we follow restriction enzyme. Here we can see here restriction enzymes. Restriction enzyme. So what is the function of restriction enzyme?
Breakdown of molecule.
Right,
DNA
breakdown of gene, foreign gene. Breakdown of foreign gene into pieces like this. This is desired gene, this is desired gene, this is desired gene. Right? So anyway, after separated this desired gene, what is the next step? Selected the vector, selected proper vector. So the vector cell is the bacteria. From the bacteria cell, the plasmids, only plasmids separated. This plasmid act as a vector. Plasmids act as a vector. Just wait. Right, where we are? Hello?
separation of plasmid.
Right.
Plasmid.
Right. So this is this round shaped part. This is called as plasmid. So here the plasmid act as a vector. Now desired gene separated. Actually, it is not a desired first. First of all, we separated the gene after break into number of pieces. In this number of pieces, we have to select the what are the desired gene later by hybridization method. So here no need to discuss about hybridization method, what is it is, but just follow. After break into number of pieces, we injected these pieces into plasmids. So here this is the one break, one piece of separated and DNA also separated. Here remember this DNA also separate with help of restriction enzyme cut into small portion. This is this portion separate from the plasmid. And later this place fulfilled with genes. Fulfilled with what you separated the genes with help of these genes. After selected desired gene, here desired gene only inserted into the plasmid. Now in this actually green, right. So blue color, are you able to see this blue color?
Yes, sir.
Right. So what is this here? So blue color means here desirable gene, right? So rest of the portion plasmid. Plasmid nothing but gene, and from the donor cell also gene. So how many genes are present in?
Two different genes.
Two different genes. So is this recombination or not?
Recombination only.
Right, recombination. So okay, you separated the gene and cut the gene with help of restriction enzyme. Now the both foreign DNA, foreign genes are joined together
Becoming
with help of ligase enzyme. Remember, cut by restriction enzyme and joined by ligase enzyme. Okay, clear? So anyway, recombinant DNA formed. This is called recombinant DNA. What are present in recombinant DNA? In recombinant DNA, foreign gene from donor cell, plasmid from bacterial cell, right?
Yes, sir.
Clear?
Yes, sir.
Now, what is the next step? Hello? What is the
Again,
fourth one?
both are introduced into the another bacterium or host.
Right. Another bacterial cell. So, because what is the natural habitat of plasmid?
Sir,
Where are the plasmids?
plasmids are inside the bacterium only.
Inside the bacteria. Okay, this is introduced into the bacterial cells. That
Yes,
is
sir.
competent host bacterial cell.
Yes, sir.
Why we introduced bacterial cell only? What is the next step? After introduced into bacterial cell, what is the next step? Next stage?
Multiplication of the desired
Right,
gene.
multiplication of rDNA. How the rDNA multiply? It can multiply their own?
No, sir.
No. Inside the bacteria, it can multiply because along with the bacterial cell, here we can observe.
Foreign DNA.
Right. So this is rDNA. rDNA. So this bacterial cell called as transformed cells. Transformed cells. Now along with the bacteria, along the bacteria division, this rDNA also divided into number of times, so that we get number of rDNAs.
Yes, sir.
Right? What is the next step after get number of rDNA? What is the next step?
We have to select our desired
Selected
gene.
rDNA by hybridization method. That is the long process. No need to discuss about that. After selected rDNAs, these rDNAs, next one, transfer of rDNA into target host cells. Right?
Yes, sir. Yes, sir.
Target host cells. So anyway, after formation clones means nothing but multiplication of rDNA. Simple. The multiplication of rDNA called as clones.
Okay.
Now this cloned engineered in bacteria and medium. Okay. Next, actually now here from the target cell. That means transfer into
Another target.
target cell. In target cell, what will happen? In target cell, apart from the rDNA, their own DNA also present, right?
Yes, sir.
But during protein synthesis, apart from the normal own DNA, this rDNA also undergoes to translation process.
Yes, sir.
Right?
Yes, sir.
What is transcription and translation?
Production of mRNA and then protein.
Okay, from the DNA, RNA prepared, from RNA protein.
Protein synthesized.
What is the main concept of the protein? After formation protein, this protein acts as an enzyme for produce, for express. What is gene expression? Already told you in offline class. Gene regulation completed. Gene expression completed. Gene regulation, protein synthesis. Gene expression means how the one character expressed, how the character repressed. Already I have given the best example.
Yes, sir.
Plant flowering example.
Yes, sir. Yes, sir.
After seed germination, immediately flower produced. Is it possible?
No, sir.
No. When the flowers are produced, then when flowers are produced in plants,
After fertilization.
where the fertilization takes place? Fertilization takes place inside the flower. How they are produced after fertilization?
Ovule. Ovary.
First after germination of seeds, small plantlets, they are developed into vegetative body only, not reproductive. When reach maturity stage, later produces the flower.
Yes, sir.
Then where are the flowering gene? Are you introduced it? Before flowering, are you introduced?
No, sir.
Itself.
Already existed in cell. Already existed in plant cell, right?
These are
Yes,
But
expressed
not express the character.
sir.
Already clearly explained in offline classes. Okay?
Yes, sir.
Right, remember. So finally, this rDNA undergoes to gene regulation. That means DNA to RNA, RNA to protein. Then only the desired character expressed in plants.
Okay,
Desired
sir.
character expressed in plants. Is it clear now?
Okay, sir.
This
Now
techniques okay?
Okay, sir.
Right. I will send this all pictures later. But anyway, you follow.
Okay, sir.
Right? Clear rDNA techniques. Hello.
Sir.
Is it clear?
Okay, sir. Okay, sir.
Right. Now in rDNA technology, two methods, vector mediated transformation and non-vector mediated transformation. Non-vector, nothing but a direct transformation. Let's see. In our books also given only vector mediated transformation, one line. Non-vector mediated transformation, just 2 lines, 2 paras. This is not enough to write the long essay questions. Right? So the genetic engineering techniques, it is carried out by two methods. One is vector mediated transformation. Vector mediated transformation, non-vector mediated transformation. Let's see the first vector mediated transformation. Vector mediated transformation here given the best example, Agrobacterium tumefaciens. Agrobacter tumefaciens. Agrobacter, where is the Agrobacter? Just wait. Agrobacter tumefaciens. Right. Are you able to see this diagram?
Yes, sir.
Yes, sir.
Right. So Agrobacter tumefaciens. This is direct method. Vector mediated method. Let's see what happened in vector mediated method. This way. Right. Gene transfer method here. First one. Vector mediated. Here the vector is Agrobacterium tumefaciens. In plants, among the various methods used for gene transfer, the Agrobacterium vector has been widely used. Agrobacterium widely used. And this Agrobacterium called as natural genetic engineers. These are called as, write down, natural genetic engineer. Natural genetic engineer. Right? From agrobacterium to M.Sc, tDNA plasmid separated. This is tDNA plasmid. The round shaped tDNA. tDNA. And this is the plant. Desired gene plants. You separated desired gene. This desired gene injected into agrobacterium tDNA or Ti plasmid. Ti plasmid or tDNA. Ti plasmid. Now it is rDNA. Now it is recombinant DNA. That is desired gene from one plant, A plant. This is A plant. Gene injected into the Ti plasmid of bacteria, agrobacterium. Now this agrobacterium, naturally, you have to know about agrobacterium. This agrobacterium, soil-borne bacteria. It is present in soil, and the nature of agrobacterium, they make a wounds to the plant's root. Through the roots, this is injected into plant body. Now this bacteria live at the another plant root. What will happen? This rDNA bacterium, rDNA containing agrobacterium, naturally grows in, enter into the plant through the roots. Later, this rDNA incorporated into the another plant DNA. And already in rDNA, the desired gene also present. Now this desired gene multiply inside the plant body. This plant, B plant body. As a result, it produces number of DNA, number of RNA, number of protein, and also express the character. Which character expressed in B plant?
Flowering.
Desired character.
Desired character. Desired character expressed in another plant. Is it clear? This is vector mediated. What is the vector here? The vector is Ti plasmid. Vector is Ti plasmid. Okay. Is it clear? Actually, the agrobacterium calls crown gall tumors. Crown gall means when it is infected the healthy plant, automatically the auxin cytokinins are released. Auxin cytokinins are phytohormones. The phytohormones are, as the phytohormones released, what is the function of auxin cytokinins? Mainly involved for cell division. They enhance the cell division. As a result, number of cells are formed as a group. In this number of cells, apart from this group of cells, this rDNA containing agrobacterium also grows. Like this, number of agrobacteriums are formed. Actually, first one agrobacterium enter into the plant. Later, immediately, due to the activity of cytokinin auxins, number of they can produce number of agrobacteriums. Right? Now, automatically, the desired gene also incorporated into the plants. Is it clear?
Is this happen naturally?
Yes, that's why it is called natural genetic engineers. It is naturally occurs. That means here only naturally occurs means naturally the agrobacterium tend to be infect the plants, most of the dicots. But we have to inject the desired gene. We have to inject a desired gene to the Ti plasmid of agrobacterium. But that is not a general. It is a general mechanism. Only agrobacterium, the main character of agrobacterium, it is frequently infected the plant roots.
Symbiosis.
Hello?
Symbiosis. Is it also known as symbiosis?
What is symbiosis first?
Nitrogen absorption. Okay, continue.
In symbiosis, both of the organisms get benefits.
Living together. Oh.
But here only agrobacterium get benefits. That means initial stage before injected DNA, desired DNA, the only agrobacterium cause the crown gall. Crown gall means that is tumors. It is the form the tumors in plant body. So that is the nature of agrobacterium. But before that, we injected the desired gene into the bacterium. Now it is entered into the plant cell. Apart from the crown gall, the desired gene also incorporated to the host plants. As a result, desired characters obtained in host plants, but not immediate effect. It takes some time. Is it clear? Hello?
Yes.
Any doubt in this?
No.
Right. So this is vector mediated. What is another one?
Non-vector mediated.
Right, non-vector mediated. Non-vector mediated in our book given only the gene gun method, and another one is DNA absorption, cell place DNA shock, electroporation. Right. The first one, let's see the gene gun method. What is gene gun? So this is gene gun method. In gene gun method, this is the one plant. Separated the desired gene from the plant. This gene encapsulated with help of tungsten and gold particle. The blue color line, tungsten and gold particles. Tungsten. Tungsten layer, gold. Tungsten layer and gold. So first in this method, the foreign DNA containing the gene to be transferred by coating. So first of all, we have to select the foreign DNA, foreign gene. This is foreign gene, right? So this foreign gene coated out surface gold or tungsten particles. Okay. Now these genes, these are all genes, coated genes. Now these are all coated genes. And injected into operators. This is called gene gun operator. After that, directly this gene gun, when you press the button, immediately ejected out and forcefully touch the target tissue. This DNA coated or gold coated particles directly touch the plant tissue or target tissue. As a result, what happened? Hello?
Yes, sir.
All the genes will enters into
The gene
the
enters into the tissue.
Yes, gene enter into the target tissue. So this technique was first developed by Professor Stanford. Who is he? Professor Stanford. Professor Stanford and co-workers of Cornell University, USA. Cornell University, USA. Right? Is it clear? Is this any mediator? In this involved any mediator to carry the gene?
No.
That means any vector.
No
Vector.
vector.
Directly we selected the desired gene. This gene coated with help of tungsten or gold particle and with help of the gene gun ejected out. And this technique is also known as particle bombardment. Particle bombardment. Let me write here. Particle bombardment. Particle bombardment or particle gun method. Particle gun method or biolistic process. Another name? Biolistic process. Biolistic process. Is it okay?
Particle gun.
okay, sir.
Right. Next one. What is given in our book? Electroporation.
Page number, please.
Sir.
238. Electroporation method. Write down electroporation method. Second one. Electroporation. Electroporation method. So in this method, foreign DNA introduced into living cells by exposure of brief electric pulse. Already we explained in protoplast culture. In protoplast culture, with help of electrodes, AC, DC current, it joined the two protoplast, right?
Yes, sir.
In the same way here also, introducing of foreign DNA into living cells by exposure of brief electric pulse. This process known as electroporation. In this technique, mixture of containing cells, here, for example, this is the cells, different of cells, and desired genes, the mixture of desired gene, right? Now apply the electric pulse. apply the electric pulse. Due to exposing of electric pulse, due to high voltage electric pulse, here the voltage between 4,000 to 8,000 volts. Very brief times, the cell absorb the DNA, desired DNA with help of the forcible action of electrical pulse. DNA seems to enter inside the cell and the nucleus. First, this is the cell. DNA entered the cell. This is nucleus. Next, enter the nucleus. Is it clear? Hello? So anyway, now where is desired gene inside the cell? First of all, you selected some of the cell, another plant cell, and you already applied the electrodes, some of the desired genes. These both are mixture. In this mixture, when you apply the electric current to the electric pulse, this desired gene enter inside the cell, later inside the nucleus. But after that, no, you selected these all transferred gene cells, separated and follow the tissue culture process. Culturing, now you have to culturing this. Only electroporation process, you follow tissue culture method. Is it clear? You know what is the tissue culturing techniques also. Is it clear?
Yes, sir.
Right. So this is electroporation. And another method not given in our book, that is micro injection method. Micro injection method. Micro injection method. Is it clear? Micro injection method. Hello?
Sir. Visible, sir.
Right. First of all, you selected desired gene. This desired gene taken into micro injection. This is micro injection with contain desired gene. Now, do you want to inject it into another target cell? This is target cell. Hold the target cell with help of pipette. This is called pipette. And remember, in this technique, target cell is positioned under the microscope. And DNA is then directly injected into the cytoplasm. Injected into, this is cytoplasm, red color portion. Injected into cytoplasm or the nucleus with help of the micro injection. So you can inject cytoplasm or indirectly into the nucleus also. This is blue color portion, the blue color one called as nucleus, red color cytoplasm. Already told you the difference between cytoplasm and nucleus, protoplasm. What is protoplasm? Hello? What is protoplasm?
Fluid in the nucleus.
What? With nucleus, this entire portion, this entire called as protoplasm. What is
Protoplasm.
cytoplasm without nucleus? Without nucleus, rest of the portion, that means only red color portion, cytoplasm.
Yes, sir.
You can inject the desired gene into the cytoplasm or direct DNA. After that, this target gene mixed with desired gene after injected. Next, these rDNA cells, rDNA containing cells transfer to the agarose nutrient medium. Agarose nutrient medium. Now follow protoplast culture, like a protoplast culture method. But in protoplast culture method, two different separated protoplast culturing. Two different, only one protoplasm separated, that is this protoplasm already present desired gene. Now the desired gene protoplasm culturing on agarose nutrient medium. Clear? Any doubt in this? Micro injection, this method called as
Injection have only desired genes, sir.
right.
What is agarose nutrient?
Did you hear last class?
No, sir, sorry.
Did not you attend?
No, now is I'm attending. Okay, let it.
Anyone explain?
Sir,
Anyone explain what is agarose nutrient medium?
Agarose nutrient medium containing all the nutrients, calcium, magnesium.
Which are useful for the growth of a
sir, it is the
plant.
growth medium.
Right. Is it clear? Hello? Who is asking the question?
Yes, sir, yes, sir.
Right. So write down this micro injection. Micro injection method. So this method also completed. Now next one. Write down fourth one. So first gene gun method completed. Electroporation completed. Micro injection completed, right?
Yes, sir.
Now liposome mediated transformation. Liposome. Right. Next one. Liposome. Liposome mediated transformation. Liposome mediated transformation. So in this method, liposomes are created artificially These are lipid vesicles created artificially, which is produced by phospholipids, made up of phospholipids. Now DNA enters the protoplast via endocytosis. Lipospores, genes or proteins to be delivered. This is desired gene. Liposome, already liposome made up of phospholipids. What is the phospholipid? Already told you the phospholipids are made up of, plasma membrane made up of phospholipids. What is the main function of phospholipids? This is phospholipids involved in endoosmosis and engulfation and transport of ions. So these are the different functions carried out by plasma membrane. So endocytosis, one of the function. So for this, we prepared artificially plasma membrane. This artificial plasma membrane called as liposome. And now after formation liposome, you inserted the desired gene inside the liposome. What is due to this activity of the liposome, this is target cell. This is target cell. When you arrange closely to the target cell, to activity of endocytosis. What is endocytosis? What is endo?
Inside cell.
Inside. This enter inside the target cell. That is target DNA enter inside the target cell. Is it clear?
Yes, sir.
Next, when it is injected into target cell, automatically it is multiply in host cell. Later, express the character. Okay. Liposome mediated transformation clear? Hello?
Yes, sir.
So mediated, non-mediated, completed. Mediated only one method, that is agrobacterium TI plasmid. Non-mediated 4 gene gun method, electroporation, microinjection, liposome mediated transformation. Clear?
సార్ లిపోజోమ్ ఫాస్ఫోలిపిడ్స్ తో క్రియేట్ చేశాక దాంట్లో డిఎన్ఏ ఎంటర్ చేస్తారు కదా
Yes, yes.
జీన్ ఓకే థ్యాంక్ యూ
Desired gene.
ఓకే థ్యాంక్ యూ సార్
Right. Next one. Now let's see some advantages, disadvantages of the transgenic plants. So before that, so anyway, in any method, either mediated, vector mediated, non-vector mediated, the desired gene entered the target cell. Later, the series of events must occur. That means whole genetical material inserted into the target cell. Later, this modified the plant, that is modified the characters. Finally, this character expressed out. With help of desired gene, finally the character expressed. Right. So let's see what are the achievements and disadvantages of transgenic plants. In general, you tell me what are the benefits.
We can
general
produce our
benefits.
better genes. We can produce our better quality genes.
Better quality.
desired qualities
Desired
of
qualities
plants.
plants, improved yields with lower need of expensive pesticides. Page number 240. Page number 240. Some benefits. 241. Sorry. Page number 241. Clearly given the benefits. Lower crop losses due to pest and weeds.
Less labor and machinery.
Less labor and machinery. more nutrition, more yields, more production. And these organic plants are adapted to extreme condition, floods and droughts. And apart from the high resistance power, we will discuss later in page number 244 because before that, let's see some of the global research activity on transgenic plants. Global research activities and transgenic plants. Let's see. Two, three. European. Right. Is it visible to you? Some global research activity and transgenic plants. So far, hello?
Yes, sir. Kanipistundi.
Right. Right. So far, what type of plants we get through transgenic technique? Let's see. Some field crops. Field crops. Sorry. Field crops, vegetables, fruits, and other crops. For the field crops, so far we obtained soya bean and cotton, maize, and canola. I don't know what type of plant, what type of crop it is. It is only two countries, Canada and USA. So these are the male field crops. The regulator approved by regulator approved of soya bean by Mexico. So that means clinical trial, because soya bean, your regulator approval means the government give a clinical trial for the experiment. Actually, what is clinical trial? You produce one transgenic plant. Can we use directly without approval?
No, sir. We have to
Without
take it.
clinical approval? We should not use.
It should be
Right.
approved.
So first of all, we will have to take the permission from the government of the country. That is called regulatory approval. So the soya bean regulator approval given by Mexico country, cotton in Canada and Egypt, maize East Europe and Honduras, canola Egypt, sugar beet Canada, USA, flax seeds in Canada and USA. These are not important in theory, but these are important in competitive level. Okay, no need to underfill this column, no need to explain. Total number of crops. I did not see the current transgenic plants, how many. I request you please search in Google how many transgenic plants so far. Find out the scientist. Now apart from the vegetables also given, the tomato, squash, pepper, a potato approval by USA, Canada, tomato USA, squash Canada. 16 crops in 40 countries, sorry, 50 countries. Fruits, papaya in Hawaii. But the regulator approval received by Canada, USA. Tobacco in USA. Chicory, regulator approval received by Canada. So these are the transgenic plants approval. in different countries, right? But this table not for theory exam. This is only for competitive exam, right? Next one, some other achievements. Let's see other achievements. Write down one by one. Benefits or achievements, applications. Transgenic plants. First, improved nutritional quality. Improved nutritional quality. Improved nutritional quality. The best example, golden rice. Best example, golden rice. In general, we see in natural rice, what happened in natural rice? Beta carotene loss. But due to the activity of milling. So this milled rice, the loss, the husk, that is layer, extra layer, the outer layer lost. The beta carotene, the in outer layer, beta carotene present and B complex also present. This beta carotene is a precursor of vitamin A, right? Beta carotene, precursors of vitamin A. If this beta carotene loss in the form of husk, that is outer layer of grain, rice grain, automatically there is no production of vitamin A. As a result, in India, generally light blindness formed. That means eye diseases are generally found. To overcome this problem, golden rice produced through the transgenic technique. Golden rice are produced through the transgenic techniques. Is it useful to us or not?
Useful only, sir.
Right.
Yes.
Now next one, can I rough it?
Okay, sir.
Next one, insect resistance.
సార్ ఒక చిన్న డౌట్. ఇప్పుడు మనం నార్మల్ గా తినేది కూడా గోల్డెన్ రైసేనా? కాదు కదా?
కాదు.
ఓకే. మళ్ళీ ఇప్పుడు మనం తినే ఫుడ్ యాక్చువల్లీ వైటమిన్ డిఫిషియన్సీ ఐ మీన్ తక్కువ ఉందనే కదా?
అవును, ఎగ్జాక్ట్లీ.
మనం ఓకే ఓకే. థ్యాంక్ యూ సార్.
నెక్స్ట్, ఇన్సెక్ట్ రెసిస్టెన్స్. సెకండ్ వన్, insect resistance. Best example, BT cotton. Bacillus, B for Bacillus thuringiensis. Bacillus thuringiensis is a bacterium. There is a pathogen for number of insect pens. Most probably we know the cotton generally infected with what is that? Caterpillar, right?
Yes, sir.
So that insect killed by Bacillus thuringiensis bacteria. So that from the Bacillus thuringiensis bacteria, we separated the gene and this gene transferred into the cotton. Now this is called BT cotton. What happens if the insect, caterpillar insect, eat the cotton leaf?
The protein present in this
Which gene present in cotton after introduced BT gene?
Protein, some sort of
Okay,
poisonous protein is
finally,
present
this
in.
BT gene produces the protein. This protein harmful to the insects,
Yes, sir.
right?
Okay.
So this is insect
Yes.
resistance. And third one, are you in page number 244?
Yes,
Hello?
sir.
No, sir.
Please turn on page number 244.
Disease resistance.
Right, this is resistance. The
Okay,
disease resistance, best example, tomato plant resistance to tobacco mosaic virus. Right? So
yes, sir.
after transfer of the gene, most of the tomatoes resistant to the TMV. So this is another benefit. And fourth, herbicide resistance. Hello?
టీఎంవీలోకి డిజైర్డ్ జీన్ ని ఇంట్రడ్యూస్ చేసినప్పుడు దాని హార్మ్నెస్ ఉండదా అంతేనా సార్?
అంతే. అంటే TMV, ask again.
టీఎంవీలోకి మన డిజైర్డ్ రికామినెంట్ జీన్ ని ఇంట్రడ్యూస్ చేసి దాని ప్లాంట్ లోకి ఇంట్రడ్యూస్ చేస్తారా అంటున్నాను సార్.
No, actually, tobacco mosaic virus. First of all, we have to find out the desired gene that is opposite character to the virus.
Yes, sir. Yes, sir.
After that, we have to select that virus and this desired gene injected into target gene.
Okay, sir.
A target cell. Later,
Okay.
this get the desired character in the form of disease resistance to the mosaic virus.
Okay, sir.
Next, herbicide resistance. In natural, we see, we must have seen the number of herbs, weed plants in crops, right?
Yes, sir.
Number of weeds are present. To kill the weeds, we generally use 2,4-D, some herbicides, right?
Yes, sir.
But alternatives are available to damage, they may damage crops as well as weeds growing in it. However, the genes from resistance to some newer herbicides have been introduced into some crop plants. For example, have you observed around the Parthenium, Congress? Do you know Congress weed?
Don't know, sir.
Yes, sir.
Congress weed, Parthenium histoporus.
Vayaribama.
Yes, we know that.
Right. Whenever you observed around the Congress weed, some other grasses, other
No,
plants.
sir.
Like that, few plants are there
Okay.
to reduce herbs, weeds. Are you clear?
Yes, sir. చెప్పండి sir.
For example, here the cotton crops or any other pulses or rice. When you cultivate, when you planting the rice plant or cotton plant, in general, before use any herbicide, apart from the cotton, some other weeds also grown.
Yes, sir.
These are aggressively grown more than
Yes, sir.
crop plants.
Yes, sir.
What if they grown like that?
The crop yield may decreases.
Right. Crop
Nutrient
yield
deficiency
decreased.
to the main crop.
Right. That's why in general we use some of the herbicides. When we use some herbicides, it is that mainly it is made up of chemical. This chemical harms to the soil and harms to the soil organisms and also plants and also farmers. That's why first of all we have to select which plant resistant to the herbs. Actually, we don't want that plant. It is not utilized for the crop. For example, concrete switch. Is it useful for crop? Is it produce
No.
vegetables? Is it useful in commercial?
To plants.
We have to select that type of plant, and we separated the gene and injected into cotton. That means I am giving some example. When you injected this gene into the cotton, automatically it has the characters herbicide resistance. Right. And now the surrounding plant, in surrounding of the plants, no herbicides are grown.
Yes, sir.
Okay.
And fifth one, salt tolerance.
Yes.
Can you harvested rice or cotton or wheat, any other crops in saline water, sea water?
Impossible, sir.
Impossible. But we must have seen some of the plants that are grown in the saline water, sea water.
Yes, sir.
We have to separate the gene from the saline water plants.
Yes.
Because already, for example, one plant grown inside the sea water. What does that mean? It can grow in sea water. That means it resistant to saline water, sea water.
Tomato plant.
Already that character, resistance character already existed in seawater plant.
Yes, sir.
Now we separated the desired gene, and this desired gene injected into the target genes, target plants,
Okay.
so that we get salt tolerance.
Yes, sir.
In this process, the University of California, Davis campus have created transgenic tomato plants.
Salt
Plant
tolerance.
grown go saline soils,
Okay.
right?
Yes, sir.
But the saline soil already the rich of sodium. The rich of sodium benefit or harmful to us?
Harmful somewhat.
Yes, harmful. But in dramatically, this sodium, excess of sodium in the vacuole of leaf cells only, but not fruit.
Okay, sir.
Okay.
Only the sodium accumulated in leaf cells. Apart
Only
from the
this,
leaf,
sir.
stem, roots also present, but not in fruit.
Yes.
So this tomato as a normal
Build up in
cultivated, look like a normal cultivated tomato.
Okay.
Right, and another one, terminator gene. The terminator gene, the transgenes introduced into the crop plants make them produce sterile seeds.
Sterile. Okay.
Sterile seeds means they cannot produce the seedlings.
New plant.
We can get plants only. Of course, seeds also produce, but they are not fertile. That
Yes,
means
sir.
if you cultivated transgenic plants, if any taxons are present, these taxons must be stored inside the seeds. What if these taxon seeds planted next year?
No growth.
Which plant growth? Which plant grown?
Harmful.
Poisonous.
Right, poisonous plants. That's why introduced terminator genes. This terminator genes ends the character of seedling. Terminate the character of seedling. That's why it produces only sterile seeds. Okay, that is only the main concept. Keep the next generation seedlings. Right?
సార్ ఇప్పుడు ఆ సీడ్లింగ్ అది స్టెరైల్ సీడ్స్ ని ప్రొడ్యూస్ చేస్తది కదా, అది సీడ్లింగ్స్ లాగా గ్రో అవ్వదు కదా,
కాదు.
సో నెక్స్ట్ జనరేషన్ ఉండదు.
ఎందుకు
ఓకే
సార్
అంటే దాంతో యూజ్ ఎందుకు ఏముంటుంది అసలు అర్థం కాలేదు.
Read next line continuation.
ఓకే
Because the toxin does not harm the growing plant.
the growing plant but only develops its developing seeds. So normally except that its seeds are sterile. Rejected by developing countries since farmers want to able to save some seeds from their
Seeds from
crop
their
to
crop
plant
plants next season.
Okay.
Here the toxin does not harm the growing plant, but only its developing seeds.
ఓకే
Only that means toxins developing inside the seeds, but not the entire plants. Already in developing seeds, if the toxins are present,
like sodium plant, they'll store all the sodium will store in
Right.
leaves. Like
Right,
that,
right.
it will seeds.
right.
Aha, okay.
The next seventh one, transgenes encoding antisense DNA. Sorry, antisense RNA. Through this process, you get some transgenic tomatoes, flowers over tomatoes. Antisense RNA complement already we discussed in first. The first of the transgenic plants is the tomato, right? Which is developed in 1994 by US. Longer shelf life. Hello?
Yes, sir.
Long shelf life.
Yes, sir.
What is the ripening hormone?
Ethylene.
Ethylene.
The same here, the antisense RNA. Antisense RNA means antisense RNA delay the production of ethylene.
Yes, sir.
Simple, delay production of ethylene. If ethylene production delay, no ripening, it gives more shelf life period. That's it. And last one, biopharmaceuticals. What is pharmaceuticals? Drugs. Bio means? Bio means
Bio
living,
and
from the living
organic
things,
living
without chemicals from the living organisms, drugs are prepared. That is called biopharmaceutical. These are in the form of proteins, or we can utilize the blood of the animals also as a drug. So anyway, remember, biopharmaceuticals means the drugs prepared from the living organism. That's it.
living organisms. Okay,
Okay.
sir.
We will continue later.
Okay,
Already
sir.
9:31.
Okay.
Thank you,
And
sir.
completely opposite the risk and controversy. Next was what? 247. Risks. What are
Okay.
the risks? Exactly, risk opposite to the achievements. I can give the best example. BT cotton. BT cotton kills the insect, right?
Yes, sir.
But here the target insect on caterpillar only. But this BT gene kills only caterpillar or other insect.
Other insects.
Other insect also. So that is the major disadvantage. Recently, the scientists discovered some of the honey bees are died due to the activity of BT gene, BT cotton. Like that, so there are a number of risk factors also there, and we can, if we use the transgenic plants, it is caused the allergic reaction in some people. So allergics. And another important sense, invasiveness. That means already, if you introduce the transgenic plants, what about already existed plant? Already normal rice is there, but you introduce new rice plant. If you introduce new rice plants, what about native plant? Is it existed further?
No.
No, that is so this is also a disadvantage. Allergic food formation, these are all some disadvantages. After the class, you have to read this all. If you have any doubt, ask later. We will continue next class. Thank you all.
Okay, sir. Thank you, sir.